Greeting from Cambodia to everyone. My name is Chanvorleak, and I am a head of Food Technology and Nutrition Research Unit and the Research and Innovation Center at Institute of Technology of Cambodia. And today is my great honor to give a presentation on my research topic, which is talk about pesticide residual in water and soil and biota of Tonle Sap Lake. So today I will talk a little bit about the background of the study, also the objective, methodology, result, and the summary. So not to waste more time, I would introduce the background of this study to all of you. So as today topic talk about the pesticide, I would like to introduce you what is pesticide. So pesticide is a mixture of compound or substance that is used to kill, repair, or control certain form of plant and animal life that are considered to be pests. And in today's topic, the pesticide will cover on fungicide, herbicide, and insecticides. So with the improper management of pesticide usage in Cambodia, there is also frequently a report on pesticide poisoning to both human and animal as well as wildlife. That is why it is important to study about the pesticide contamination in various type of samples. And one more aspect of the topic is considered about Tonle Sap Lake. So as you can see in this figure, Tonle Sap Lake is located in the middle of Cambodia. It functions as the heart of Cambodia. And it is also one of the biggest freshwater lake in Southeast Asia. For agricultural land, it's cover up to 42% of agricultural land in Cambodia. And according to a survey by World Bank, it is reported that more than 67% of the farmer in that area use pesticide for agricultural practice. And those pesticides eventually can contaminate it to the water of the Tonle Sap Lake. So I would like to play a video why it is important to study about Tonle Sap Lake. So please spare a few minutes to see this video.
បឹងទន្លេសាបទ្រទ្រង់ជីវភាពរស់នៅរបស់មនុស្សរាប់លាននាក់មនុស្សជាច្រើនប្រើប្រាស់ទឹកបឹងសម្រាប់ងូតធ្វើម្ហូបលាងសម្អាតចំណីអាហារនិងបរិភោគជារៀងរាល់ថ្ងៃផងដែរប៉ុន្តែតើទឹកបឹងពិតជាស្អាតនិងមានសុវត្ថិភាពមែនឬទេអ្នកស្រាវជ្រាវមកពីប្រទេសជប៉ុននិងកម្ពុជាបានសហការគ្នាធ្វើការស្រាវជ្រាវដើម្បីស្វែងយល់បន្ថែមអំពីទឹកបឹងនេះហើយបានផ្ដល់នូវមតិយោបល់ដ៏មានអត្ថប្រយោជន៍ជាច្រើនជារៀងរាល់ថ្ងៃបរិមាណទឹកសំណល់នៅក្នុងស្រុកដែលមិនបានប្រព្រឹត្តកម្មជាច្រើនបានហូរចាក់ចូលទៅក្នុងបឹងនៅក្នុងទឹកសំណល់នេះមានលាយឡំទៅដោយលាមកទឹកនោមដែលហូរចេញពីភូមិអណ្តែតទឹកក្រៅពីនេះក៏មានការបញ្ចេញទឹកសំណល់ឧស្សាហកម្មដែលមិនបានប្រព្រឹត្តកម្មព្រមទាំងជីគីមីដែលជាកាកសំណល់ពីសកម្មភាពកសិកម្មនិងវិរិវប្បកម្មផងដែរដូច្នេះនៅក្នុងទឹកបឹងមានផ្ទុកបាក់តេរី
So as you can see in the video, Tonle Sap Lake hosts a great deal of floating village that reside on the lake, and the people there, they use the water directly from the lake for everyday purpose like cooking, washing, and yes, drinking as well. And For the topic of today, the study area is not covered around the Tonle Sap Lake, but in the study area will be focused on Chhnok Tru, which is located around here. Which is located here. It's the connection between Tonle Sap Great Lake and Tonle Sap rivers. So the special characteristic of Chhnok Tru area is that it is covered by water in rainy season, and during the dry season, there is a lot of available land for agriculture. It is why in that area, the farmer practice a lot of agricultural activity like rice field or vegetable farmings. So the objective of this study is we want to study about the pesticide contamination in 4 types of sample, which include water, vegetable, fish, and farm soil. That was collected from Chhnok Tru area during 2018 and 2019. So the sample will be scanned for pesticide compound, and also as well as it will be quantified for the 23 targeted compound. And most of the 23 targeted compound, they are the banned pesticide. It means that it does not allow to be used in Cambodia anymore. So in here, in this map, you can see the sampling area of Chhnok Tru area. So we have a total of 18 water samples that were collected, and also 8 vegetable samples, as you can see in the green sampling point. And also the soil sample was also collected from the farm that is that we collect the vegetable as well. And for the fish sample, we buy it from the farmer, from the fisherman in that area. So a total of 4 types of fish were also studied for pesticide residues. And here is the methodology for pesticide analysis in water sample, vegetable, and fish sample. So I will not go into detail onto the process of how to analyze this sample. So now please see the result of our study. First, we talk about the qualitative data, which is the scanning of pesticide in various types of sample. As you can see here, as you can see from this table, there are 16 pesticide compounds were detected in vegetable sample, and among the 16, 6 were fungicide, and 5 were herbicide, and another 5 were insecticide. And from various sample type, pH plant from farm 1 was the most contaminated with 6 pesticide compound, and including 1 banned pesticides. And Mavin 4 and disulfiram was the most frequently detected pesticide compound among all. And for fish sample, we found only 2 active compounds in fish sample, and both are fungicide, which are bipenil and fluoroconazole. And in soil sample, 10 compounds were detected, and the most detected compound is fluoroconazole, which is found in 92% of soil sample, and followed by chlorpheniramine, 30% of the sample, 32% of the sample, and oxalibyl in 22% of all sample. For water sample, there is quite a lot of compound in water sample. So from this figure, it shows about the fungicide that is detected in water sample. So in total, 9 fungicides were detected, and chloronap was the most frequently detected, followed by 2 pennyphenols. And among the 18 sampling sites, JA16 were the most contaminated one with 4 fungicide compounds. And bipenyl, fluoroconazole, and phyroquinone were also detected in vegetable sample. And for herbicide, only 2 compounds were detected. And dimethamethyl was the most frequently detected in vegetable as well. So, and next one is insecticide. So insecticide, we found 10 insecticide was detected in our water sample. And among them, 60% were banned pesticide, including BHC, chlordan, ongtrin, heptachlor, and mebendfor, and paratrans. And among all, BHC and paratrans were the most frequently detected. And J12 is the most contaminated site. And for the compound detected here, chlorpheniramine and mebendfor were also detected in vegetable sample. So let's move to the quantitative data. In vegetable sample, we found that only 2 out of the 23 targeted compounds were detected, which is methylacyl and pyroquinones. So both compounds are in the toxicity class 2. According to the WHO, it is moderately hazard. The detected concentration of pyroquinone in this study was lower than the in the sample. And both methylacyl and pyroquinone were detected in the lower than the standard limited in the vegetable samples. And actually for soy and fish sample, we did not detect any targeted compound. So we only detected in vegetable and water sample. So again, there is more pesticide compound were detected in water sample. So in total, we detected 11 of the targeted pesticide compound in water sample. And in there, among those, some also banned pesticide in Cambodia. So, and those banned pesticides were also in class 1A, which is extremely hazardous, also present in this sample. And the detected compound of chlordan, which was very high compared to the standard of the WHO in drinking water. And ongtrin and HCA were also exceeding the water quality standard for public water area of public health protection by the Ministry of Environment of Cambodia. So if we talk about the pesticide contamination, we should not focus only the individual compound, as one sample can be contaminated with more than 2 compounds. more than one compound. So the total contamination level also important to this study. From this experiment from the project, we also found that the total contamination level of pesticide residual in all water sample range from 0.09 to up as high as 26.1 microgram per liter. So this is a quite serious concern as the contamination level are very high. And according to the environmental protection agencies, the sum of pesticide contamination should be should not be over 0.5 microgram per liter. So by seeing this result, you can see that almost all the detected sites were presented with a very high concentration of pesticide compared to the standard, which is very give us a big concern, especially for the people who live in the floating area, that they use the lake water directly as drinking water. So in conclusion, the results show that 72.2% of water sample and 87.5% of vegetable sample and 100% of soil and fish sample were contaminated with at least one of pesticide compound. And in this study, we found that 16 active compounds were found in vegetable sample and 10 in soil sample, fish, and 2 compounds in fish sample, and while 21 compounds were found in water sample. And fluoroconazole was identified in every type of sample. It means that we found this compound in water, soil, fish, and vegetable sample. And the total contamination level in water range from 0.09 to 26.1 microgram per liter in water sample. And in this study, we also found some banned pesticide compounds such as HCA, glutamate, chlordan, andrin, heptachlor, levin-4, and paraxylene. And the presence of this compound can be due to the highly persistent in the environment or illegal applications. Actually, in Cambodia, pesticide formulation, pesticide usage, and disposal, as well as pesticide storage, still continue to be a big concern. And government regulation are an important part of ensuring the safety of agricultural products for consumer. Although in developing country, they have a complex system in place to control pesticide sale and use. But other countries, especially developing countries like Cambodia, we do not all adhere to the same strict rules. So precaution measure should be include like testing the safety of active compound and also performing the mandatory assessment, as well as increasing the environmental awareness and decrease the impact on the ecologies. So and here is the project funder. So this project is funded by the research grant provided by the Site and Technology Research Partnership under the SATREP projects. Here is the reference, and thank you for your kind attention.